Hello, everyone. Welcome to Foresight's Biotech Group, sponsored by 100 Plus Capital. Today, we're very lucky to have Umberto Simeoni on with us. He'll be presenting on early life conditioning of health and aging. He has served as a professor of pediatrics at Louis Pasteur University and University Hospital of Strasbourg. And his research focus is on how early life exposures ranging from nutrition to stress can program biological systems in ways that shape lifelong health. So with that being said, thank you so much, Umberto, for joining us. We're really excited for this presentation.
Thank you so much for the invitation, and hello to everyone. So this presentation is indeed on early life conditioning of health and aging. And I would propose to follow this outline. First, we may examine our current knowledge of the general concept of developmental conditioning or programming of health over the life course. Then we see what we have learned from the long-term consequences of what we call the great perinatal syndrome, which are the major complications of pregnancy and the following consequences to the offspring, and the related animal models, and finally, possible the frequency of chronic non-communicable diseases is increasing dramatically around the world, as shown here for hypertension, diabetes, and mental disorders. These diseases kill dozens of millions of people each year worldwide and induce billions of dollars costs per year in many countries. They are associated with age and they hamper healthy aging. They are mostly avoidable and preventable as they are strongly linked to lifestyles, harmful behaviors, and adverse environmental exposures. With a surprising major window of sensitivity to environmental factors during the very early stages of development, during the period called the first 1,000 days of life, that starts before conception and covers intrauterine development, birth, and the infancy. Indeed, the pioneering studies by David Barker and his group in Southampton have revealed how adverse conditions in utero and in infancy induce poor health in adulthood. They were able to find records of birth weights and living conditions during infancy from people born at least 80 years ago, and to link them to their current cardiovascular, metabolic, and mental health. They showed that low birth weight and poor weight gain up to 1 year of age were associated with an almost double risk of dying of heart attack, as well as diabetes. The concept of developmental origins of health and disease, also known as developmental programming or conditioning, was born. The key facts to be understood is that during the unique window of sensitivity constituted by early life, the effects of stimuli from the environment are not only short-term, but also persisting, lifelong, while the same stimuli applied later in life have only transient effects. This is true for poor fetal nutrition and chronic fetal stress, of which low birth weight is a surrogate, as in the Barker's hypothesis, but also for other environmental influences. Thus, timing, more than the nature of adverse stimuli, makes the difference. This is very simply shown in the first graph on the left of this slide. In this ancient study, applying a relatively benign temporary food restriction to rat pups in the middle of their growth has only a temporary, reversible effect, as shown on the graph on the right of this figure. While applying the same nutritional restriction at earlier stages of life is followed by a permanent change or reprogramming of their growth corridor compared to the controls. In the middle of the slide, we can see the impressive size of the effect of poor environment in early life, obtained with data from a major Finnish cohort. The effect of lower birth weight on the z-axis appears similar in amplitude to that of excess weight at adulthood on the x-axis, which mild low birth weight amplifies considerably up to 80% in addition to weight at adulthood. This study shows indeed that subtle stimuli in early development can have ample silent effects that reveal only later in life, as most of the birth weights in this cohort were within the normal range, between 4 and 3 kilograms. Finally, on the right of this slide, we know today that the correlation follows a U-shaped curve. This graph is on early conditioning of renal failure, and excess early nutrition and the high birth weight may have similar effects to those of a low birth weight. This recent study shows a stunning effect of one single element, sugar, in conditioning in early life the risk of later diabetes and hypertension. This study leverages quasi-experimental variations resulting from the end of the United Kingdom's sugar rationing after World War II that occurred in September 1953. In that time, the rationing restricted sugar intake to levels within current dietary guidelines, yet consumption nearly doubled immediately post-rationing. Comparing adults conceived just before or after rationing ended, the authors found that early life rationing reduced diabetes and hypertension risk by about 35 and 20% respectively, and delayed disease onset by 4 and 2 years. Protection was evident with in utero exposure and increased with postnatal sugar restriction, especially after 6 months, when solid foods likely began in the babies. In utero, sugar rationing alone accounted to about 1/3 of the risk reduction. Not only nutritional challenges are responsible for adverse early programming of biologic systems, early life psychological stress, which is known to alter health, has also long-term consequences on their lifespan and mortality. Thanks to a unique French legislation for war orphans and children of disabled soldiers, the authors of this remarkable study were able to analyze the adult mortality of individuals born between 1914 and 1916, whose fathers were killed during World War I, which exposed their mothers, then pregnant, to brutal stress when they received by mail the announcement of the death of their husband. The mean loss of adult lifespan of orphans who had lost their father before birth was 2.5 years, and was the result of increased mortality, mainly before the age of 65. Adult lifespan was not reduced when the father's death occurred after the infant's birth. On the other side, a much lower influence was observed in women compared to men. These results support the notion that intrauterine exposure to a major psychological maternal stress can affect longevity. One interpretation of these observations taken together is that development is an adaptive, predictive phenomenon which needs the signals from the early environment to fully adapt the individual to future environment. Several environmental factors that constitute the exposome between preconception and the end of infancy, resulting from lifestyle, behaviors, and toxicants, can condition health on the long term. The early life exposome consists in nutrition, stress, either physical, mental, social, or economic, physical activity, sleep, exposure to toxicants such as tobacco or endocrine disrupting compounds, and other environmental stresses. As shown by the green and red arrows, respectively, developmental conditioning or programming may lead to a healthy life course, including longevity, or to less favorable outcomes with an increased risk of all categories of chronic diseases and or accelerated aging. The development of the embryo and of the fetus involves major changes in gene expression to allow the temporal spatial development of organs and systems, as well as cell differentiation and maturation. The epigenetic imprinting is key to allow this process by controlling gene expression and translating environmental cues into specific marks that will set the reference functioning of the genes responsible for the main physiologic and biologic functions over the life course. The relationship between early life adverse events and their long-term consequences is widely considered to involve persisting specific epigenetic imprinting. This is shown here by the effects that at adulthood of early life exposure to the Dutch famine that occurred during World War II. They include increased incidence of metabolic, cardiovascular, and mental disorders. In this population, DNA methylation signatures on genes involved in growth and metabolism were differentially expressed at adulthood in subjects who were conceived during the famine in the winter that covered 1944 and 1945 compared to unexposed siblings. The consistent chronologic variations between daily caloric ratio, average temperature at conception during the winter, which are showed below in the B graph, and DNA methylation differences at adulthood are striking. In this scheme, age and the life course are represented on the x-axis, while the reduction of health capital over time is represented on the y-axis. Three examples of health trajectories influenced by behavioral and environmental factors in early life and by eventual major adverse conditions in later life as well, are shown, starting from the periconception period. They are represented above the genome in light blue here, which remains, of course, the same. According to this interpretation, even subtle environmental cues in early life, here during the first 1,000 days, may set durably the slope angle of health trajectories that may become strongly divergent later. The slope of risk to the available health capital may be favorable. This is the trajectory A in green, eventually influenced by later events on the full line, or less favorable, such as red trajectory, and or intermediate, as in the blue line. So targeting favorably the slope of risk trajectories by preventive behavioral and environmental measures in early life may result, here with the green arrow, much more efficient than acting later in life, in adulthood, of course, while all the components of the exposome during development may influence the orientation of such health trajectories. We already mentioned the components of this exposome. Also, the slope given by the environment through epigenetic mechanisms and the resulting early established epigenome may today be considered as components of the predisposition of a person to develop a chronic disease. We usually use this term when we say it's not only behavior or environment, there is a difference, and there should be a difference in the genome or in genes, a genetic difference. Indeed, there is certainly an influence of the genome, but the influence of the environment and mainly the epigenome may be also extremely important. Finally, developmental plasticity thus appears maximal during the very early stages of development and then decreases, of course, over the life course. Developmental plasticity is how one single genotype can translate into an infinity of phenotypes. Genotype can translate into an infinity of phenotypes. While the difficulties to respond to health challenges increase with aging. Finally, experimental studies have shown that epigenetic marks can be inherited by several subsequent generations, which leads to the concept of transgenerational heritability of acquired traits, as epigenetic imprinting is the translation of the influence of the environment. And this is in the next generation with the same life cycle, which starts with conception and early life and follows the life course. Favorable but also less favorable imprints may thus be transferred to the future generation. Despite the epigenetic reprogramming phases that intervene during gametogenesis and during pre-implantation embryo development, this may explain why the periconception period may be particularly sensitive to environmental factors. The resulting potential leveraging mechanism linked to the cycle of reproduction may contribute to the velocity of the current global dissemination of diseases such as obesity. Such heritability constitutes an additional argument in favor of preventive interventions targeting the beginning of life and in particular the periconception periods, thus oriented towards young adults, parents-to-be, or at school age for an effect on the next generation. Not only maternal influences matter. The role of paternal influences has been evidenced both in human and in animal studies. In this study, the authors used the mouse model of maternal stress. Male offspring showed depressive behaviors and glitches in glucose metabolism. There is often an association between behaviors, psychological consequences of stress, and the impact on the hypothalamic adrenal axis with poor glucose tolerance and altered glucose metabolism. The sperm of these rats showed abnormally, of these mice, showed abnormally high expression of microRNA, which are non-coding RNAs and part of the epigenetic mechanisms, one being linked to stress and regulation of metabolism, in particular the microRNA 375. Similar changes were observed in the F2 generation, and behavioral responses persisted in the F3 generation. Interestingly, a microRNA from maize F1 sperm injected into freshly fertilized eggs from untraumatized mice, let's say, while in a normal situation, such injection resulted in mice with comparable depressive behaviors and metabolic disturbances passed to the next generation as well. Thus, sperm responds to the environment, and as the authors stated, dad is having a much larger role in the whole process rather than just delivering his genome and being done with it. Subtle changes in sperm microRNAs set the stage for a huge plethora of other effects. So this was the first part, and I would propose you to move to the great perinatal syndromes and what we have learned from them, and as well as related animal models, which try to mimic them. The great perinatal syndromes, such as intrauterine growth restriction, preterm birth, maternal overweight or obesity, or gestational diabetes, appear in this context as particular conditions for altered developmental programming due to a complicated pregnancy. In addition to the short-term consequences of each specific complication of pregnancy, the offspring, whatever the specific syndrome, is at increased risk of chronic diseases later in life. Whether the perinatal syndrome nutritional characteristic is undernutrition, for example, in fetal growth restriction, or overnutrition, such as in the case of gestational diabetes or maternal obesity situations, the long-term consequences are very similar. Despite opposite triggering events, their mechanisms also, at the level of the systems or at cellular or molecular levels, share similar characteristics. For example, hypertension and cardiovascular disease programmed in such circumstances associate three principal mechanisms: reduced nephron endowment, altered endothelial function, and altered responses to stress. And one key cellular mechanism, which is accelerated cell senescence, whatever the starting point. On the metabolic side, impaired glucose tolerance or type 2 diabetes in the offspring at adulthood is related to reduced insulin sensitivity, insufficient insulin production, eventual obesity. And these are our common chronic diseases that we know, unfortunately, very well. This leads the great obstetrical syndromes to be sometimes considered as hard developmental origins of health and disease experiments in nature. I'm sorry, I'm trying to move to the next slide. Here we are. Animal models help understanding the intimate mechanisms at work in the major perinatal syndromes. Here, in an experimental model of maternal overnutrition in mice due to high-fat diet during gestation and lactation, the offspring developed cardiac hypertrophy and fibrosis, mediated by TGF-beta. At the epigenetic level, we observed characteristic alterations of the microRNA profile in this offspring of non-coding RNA biosynthesis, especially at the digital syndrome critical region 8. With the same model, we recently observed that maternal high-fat diet induced long-term mitochondrial alterations in the offspring heart, including lipid content and inflammation. Such mechanisms are likely to underlie chronic heart disease in general and accelerated aging of the heart related to poor nutrition and lifestyle in early life. Moving to humans, and especially the situation of preterm infants and their long-term outcomes, as well as their endothelial progenitor cells, which are involved in angiogenesis and vasculogenesis. Altered endothelial progenitor cells numbers and function were associated with increased cell senescence in correlation with decreasing gestational age and a related sirtuin 1 deficiency with altered histone 3 K9 methylation. Of note, this profile may pave the way for later hypertension, which is a known consequence of being born preterm at adulthood. A similar condition has been identified by others in opposite nutritional conditions, no low birth weight, but after exposure to maternal gestational diabetes. So different triggering situations, but common mechanisms, especially in terms of accelerated cell senescence, which is mainly a stress-induced cells. In addition, interestingly, resveratrol treatment restores cell proliferation by increasing sirtuin-1 expression at the protein and RNA levels in preterm endothelial colony-forming cell, which means endothelial progenitor cells. So it is possible to reverse these problems, at least in vitro. The endpoint mechanism of altered developmental programming, which seemed to be accelerated stress-induced senescence, is thus found in many studied organ systems, whatever the initial triggering factor, under or overnutrition, in humans as well as in other species. As other examples, we observed accelerated senescence also in developing liver exposed to overnutrition by litter size in a rat model with glucose intolerance. In the same IUGR rat model, we observed also that the kidney displayed a similar sirtuin-1 deficiency and accelerated cell senescence. This deficit in sirtuin-1, which is a protective protein against cell senescence with an epigenetically altered methylation of histone 3 related to the SIRT gene, is a common feature to all the study situations in our experience as well as in that of others. Of note, such characteristics persisted in adult offspring, even.
Oh no, I think he just dropped off. Hopefully he'll be back soon.
So here we are. Well, just at the end of this review of the common mechanisms that we learned from the great obstetrical syndromes and animal models we used in our research, I would like to describe briefly this extremely interesting study which has been performed about the issue of whether familial variations across the lifespan are genetic or environmental or both. Of course, this is the fundamental issue in these experiments, and it's between the innate and the acquired. And this study is extremely interesting because the role of environmental causes of DNA age variation, methylated DNA variation across the lifespan has been explored by pooled genome-wide DNA methylation data for more than 4,000 people aged 0-92 years from 1,800 families. DNA M age was calculated using the Horvath epigenetic clock. For all pairs of relatives, their correlations increased with the time spent living together, especially in twins, which are part of the subjects who participated in this study, and either monozygotic or dizygotic twins. For correlations then decreased with time spent living apart at similar rates between monozygotic pairs and dizygotic or sibling rates. These persisting patterns were best explained by cohabitation-dependent shared environmental factors and genetic factors, even when analyzing parent-offspring pairs, for example, or spouse pairs. Genetic factors explained only 13% of variation. Many of us are convinced that environment is a key point in aging and chronic diseases as well on a genetic basis. But this is an extremely important study, in my opinion, that confirms the importance of the role of the environment and of methylation of DNA in the epigenetic mechanism. The authors showed similar results using two other epigenetic clocks, suggesting that the observations are robust to how DNA age is measured. And in addition, results for the other clocks were consistent with there also being a role for prenatal environmental factors in determining their variation. An additional conclusion of this study is that the equal environment assumption of the classic twin study might not hold for epigenetic aging. Finally, about possible interventions in this field. Economic models show that the rates of return to investment in human capital increase considerably at the timing of preventive intervention targets earlier ages, down to conception, pregnancy, conception, infancy, childhood, schooling, up to job training. Many experts today consider that the pre and the periconception periods are the most sensitive to the environment in terms of normal or altered developmental programming. Early prevention health policies may thus be most effective in reducing the burden of chronic non-communicable diseases and aging, and promoting a better global health in its three dimensions, somatic, psychic, and social, when targeting younger ages and especially the early stages of life. Yeah. Such economic advantages have led public or private organizations in various countries to adopt public health preventive policies, especially programs of early prevention that target the first 1,000 days in the population. Such programs aim at supporting and guiding parents to be in preconception, adolescents as well, and possibly children in schools, based on lifestyle, environmental exposure, and social economic support. Suboptimal or unhealthy behavioral and lifestyle factors, such as poor nutrition, smoking, excessive alcohol intake, stress, are associated with epigenetic marks and epigenetic age acceleration. Reprogramming interventions through improved lifestyle, reducing adverse environmental exposures and stress can counteract the development of age-related pathologies. But to be effective, interventions and support ideally should meet certainly well-recognized criteria. They should be personalized and include partners when possible. They should be participatory, with the patient being considered an actor in his or her own stress reduction, for example. They should be structured in time and content, supportive and sustained, with the possibility of autonomy based on continuous feedback. Possibly preventive, starting very early from the preconception period, and of course, evidence-based. This means that teamwork is generally necessary, allowing for interprofessional, nutritional, psychological, and socio-economic approaches, and sufficient time dedicated to each patient. Lifestyle medicine appears to be an important part of this support. Also, e-health and m-health systems associated with in-person or AI support may help simplify these approaches, which are quite heavy, but make a lot of sense. On the other hand, another kind of reprogramming is proposed. To extend lifespan, it is essential to unveil molecular mechanisms involved in aging. As for healthspan, a better understanding of the mechanisms involved in age-related pathologies is crucial. But the reversibility of the epigenetic marks makes them promising targets for rejuvenation. In vitro studies have demonstrated that cellular reprogramming to pluripotency reverses cellular age, but alteration of the aging process through reprogramming has not been directly demonstrated in vivo. An increased risk of tumors has been evidenced in addition with such approach. So apart cellular reprogramming to pluripotency, partial reprogramming has been proposed through the short-term only cyclic expression of OSKM reprogramming factors. So you may know these factors are involved in the normal reprogramming that occurs normally during just gametogenesis and pre-implantation embryo development after fertilization. In this study by the group of Campo, cyclic expression of OSKM programming factors induced with doxycycline ameliorated cellular and physiological hallmarks of aging in mice. In addition, it prolonged lifespan in a progeroid mouse model of premature aging. You certainly know progeria, the progeria disease, which is a condition for premature aging. Similarly, the expression of OSKM in vivo improved recovery from metabolic disease and muscle injury in older wild-type mice. This is one first approach for partial reprogramming and short-term reprogramming. In this study, by the group of Vittorio Sebastiano at Stanford, the possibility of transiently express nuclear reprogramming factors that we just mentioned in a non-integrative way based on mRNA was used to counteract aging in human fibroblasts and endothelial cells, as well as in aging chondrocytes. In figures H and I, transcriptomic and epigenetic clock analysis showed more useful signatures upon transient expression of OSKM and L reprogramming factors in human fibroblasts on the left and in human endothelial cells on the right. Transient OSKM and L reprogramming factors expression also mitigated inflammatory phenotypes in diseased chondrocytes obtained from cartilage biopsies in aged patients diagnosed late-stage osteoarthritis. Healthy cells in blue compared to 8 osteoarthritis cells in red, and the transiently reprogrammed osteoarthritis cells in light blue here, with a test of cell proliferation, capacity, production of ATP, and also uptaking of reactive and spaces, all improved after such treatment. So both studies are very interesting approach. Further research is, of course, needed to understand better the mechanisms of epigenetic reprogramming of cells, especially partial reprogramming of cells. But they are absolutely promising, at least for the moment, at the cellular level. Finally, I would just mention the further developments which look possible and on which we focus. They are in 2 different fields. In the biotech and biology field, the present and the future is about developing new biomarkers and potential therapeutic targets in early screening for the prevention of chronic disease, such as DNA methylation modifiers, and especially, and this is extremely convincing in our opinion, non-coding, especially microRNAs, but also in the promotion of health through preventive intervention and policies based on lifestyle and environmental exposures, with the development of e-health, AI-supported, either public or private digital systems that allow a personalized, participatory, preventive interventions program targeting lifestyle and simply environmental exposure. So we have a very simple, common sense-based approach, and also cutting-edge biotechnological and biological approach, which are possible. And investing in such research is certainly interesting, especially when targeting the early stages of life for aging as well as for chronic diseases. In conclusion, I hope to have convinced you that early life stages environment matters for the health span, for aging, and for the lifespan, and need probably to be understood within a life cycle interventional perspective to try to promote healthy aging. And then, again, my apologies for the interruption, which was absolutely accessible to my possibilities. It was a general power problem. And apologies for this, and thank you for your time and your attention.
Thank you so much, Umberto, for joining us and presenting. Unfortunately, we don't have any time for questions, but just want to thank you again for joining and for presenting this fascinating work that you're doing. Thank you, everyone, and we will see you back here next month.